After watching the video on the protein structure, let's now take a closer look at each of the protein structure. In the primary protein structure, we are more interested in the sequence of the amino acids. As you know, all proteins are made up of the 20 essential amino acids, and these amino acids can be linked in different sequences to form different types of proteins. So in the primary structure, we know the exact sequence of each of the amino acids. In the secondary structure, we now see the presence of hydrogen bonding between the carbonyl groups of one amino acid to the amino group of the other amino acids, with no interaction between the side chains. The tertiary protein structure occurs when the interactions are present between the side chains of different amino acids. And in the quaternary protein structure, it occurs when there are more than one polypeptide chain. Now, let's look at the primary structure of protein. In the primary structure of protein, we can see that amino acids are linked to each other in a unique sequence by peptide linkages, and larger proteins can easily contain 100 or more amino acid residues. We can deduce the primary structure of proteins by using enzymes to cleave the polypeptide chain. For example, if a polypeptide chain consists of 5 amino acid fragments, we can use enzymes to cleave the peptide bonds between amino acids. For example, if we use an enzyme A, it might cleave the polypeptide chain this way, and we will get the following fragments. But if we use another enzyme, let's say this enzyme B, it might result in cleaving of the peptide bond in different positions. Then we might end up with these fragments. By piecing information from the fragments gotten by the hydrolysis reactions of different enzymes, we can deduce the structures of the polypeptide chain. After watching all the videos, there is a written assignment that you can try out to deduce the unknown structures of 2 polypeptide chains. Please try so that you understand how this can be applied in question solving. For the secondary structure of protein, there are 2 possible ways of folding it by hydrogen bonding. One way is called the alpha helix, and the second way is called the beta pleated sheet. In the alpha helix, we can see that the long polypeptide chains are folded into the shape of an alpha helix, with hydrogen bonds between the carbonyl groups of one amino acid residue and the hydrogen atom in the amino group of another amino acid at regular interval along the same polypeptide chain. We can see that the chain is held by hydrogen bonding between every 4th peptide bonds, and this hydrogen bonding stabilizes the secondary structure because it minimizes the hydrophobic sides of protein to water and maximizes the hydrophilic sides to water, increasing its solubility in water. Hydrophobic sites consist of organic functional groups that are generally non-polar in nature, whereas hydrophilic sites contains functional groups which are highly polar in nature. Let's now take a look at the beta pleated sheets. In the beta pleated sheets, we see polypeptide chains being arranged either parallel or anti-parallel to each other, and in between the sheets that consists of hydrogen bonding between the carbonyl groups of one chain to the amino groups of the adjacent chains. Below you can see the two possible forms of beta pleated sheets, the parallel beta pleated sheets as well as the anti-parallel beta pleated sheets. In order to see which is parallel and which is anti-parallel, we first have to identify the C-terminus as well as the N-terminus of each polypeptide chain. So I'm going to shade the C-terminus of the polypeptide chain in yellow. And the N-terminus in light blue. As you can see from the diagram that I've just shaded, for the parallel chain, the C-terminus and the N-terminus are in the same direction, whereas for the anti-parallel beta pleated sheets, they are in the opposite directions. This concludes our second video, and in the next video we are going to look at the tertiary as well as the quaternary structure of protein.